Good morning, everyone. Thank you, Amber, for an invitation. After Amber's talk, I don't know what to say, but congratulations again. I think this is a great progress to see the gene therapy. And I'm a senior scientist in Boston Children's Hospital in Dr. Alexander Rothenberg's lab. And as a basic scientist, it's really great to think about our work at the bench can actually go to the clinic. So I know Amber 2022, 2023, around that time, and I was in one of those circles in that in the previous meeting. Really great to see all the new friends and old friends and colleagues and also new generation. So I want to share a little bit of work that we're doing in Boston Children's. And also because of that, we want to bring it to the clinic, and we have a startup company called Galibra Neuroscience. As you can see from that, it's a little bit logo that I made. It's related to the synapse, so that's the connection between the neurons, and the neurotransmitter that we are focusing on is GABA. So you'll hear about this in this talk. So let me follow up. Yeah. So I'd like to, for today's talk, I would like to introduce some of the key challenges. Although we now know that gene therapy is a thing in SLC6A1, it might work. We want to improve it. We want to optimize it. So we want to think about back to the science and think about some of the key challenges that SLC6A1 disorder might face. And I'll review some of those current status of SLC6A1 gene therapy and our preliminary findings in our lab. So challenge one is that the brain is developing, and GABA is the major inhibitory neurotransmitter in the brain. The signaling is very important for brain development. So on the left, there's a cartoon showing a synapse. This is where a neuron communicating with each other. On the top, there is a presynaptic synapse site that releases GABA. This is gamma-aminobutyric acid. And these blue circles are the GABA that is released into the synaptic gap. And then it will bind to GABA receptor, and then to the downstream. And in terms of GABA, it signals the next neuron to keep quiet, to silence. So that's why GABA is inhibitory. SLC6A1 is encoding a protein called GAT1, which is GABA transporter. It lives in this presynaptic terminal. And its goal is to recycle this released GABA from the synaptic cleft back to the presynaptic site. So as you can imagine, in the GAT1 mutation, when SLC6A1 is mutated, GAT1 is not functional. So there's not much of recycling that can happen back to the presynaptic site. So what happens is that there's accumulation of this GABA in this synaptic cleft. It does not go anywhere, but then it turns out it will spill over to not only the synaptic site, but also to what we call the extrasynaptic site. So it just wreak havoc in the whole brain in these neuronal specific sites. And we think that there are a lot of compensatory changes that's happening during this brain development. Some of this that has been documented in the past 20 years in the basic research is that there might be tonic inhibition. So those are the level of inhibition that is constantly governing our brain. That might be enhanced because of this spillover. And there might be also some receptor internalization that compensate these changes. So gene therapy is a rather directly approach because the gene is not functional. Let's put it back. So what we think that is when we can successfully restore GABA, this GABA transporter, GAT1 function, there should be a renormalization of this GABA. That should do a few things, including a reversible of these changes that's happening before the treatment. So whether this reversibility is true is very important for a successful gene therapy. A second challenge is a therapeutic window, as we know that the brain is developing, and of course, the earlier, the better. I really like this work from Stephen Gray's work that's published last year that has done a good work and a comprehensive study of different ages of treatment of gene therapy. And I would like to draw your attention to this graph, which shows that the EEG, the epilepsy reversibility, is actually rather limited. So about like adolescence age of a mouse, that reversibility seems to be stopped. So that means the efficacy might be quite limited. So how are we going to break through that, and how are we going to design a gene therapy that might be also amenable to older patients? That could be the goal. When we think about the third challenge is that where is SLC6A1 expressed? So this is from a mouse brain, the sagittal section, so it's just a cross-section of the brain. And we're looking at the transcript, the mRNA of SLC6A1. You can see that these tiny dots are signaling the expression of this SLC6A1. You can see regional enrichment, including the hippocampus, that's the region for memory and learning. And there are also cerebellum, that is for motor function. So you can see that there are enrichment on SLC6A1 in these regions. But also, if you look at the single-cell RNA sequencing data, that tells you what cell express these genes. You can see interestingly, there's an enrichment of SLC6A1 in these what we call the interneurons. So those are the neurons that is signaling the inhibition, that release GABA. So we know that now if we want to put back the gene to a certain place, these might be our target. Another way to think about SLC6A1 is that what other cell types that might express? So I told you about that these are the one expressed, and you can see that the GET1 that is a marker for inhibitory neuron. You can see they are quite overlapping. So SLC6A1 expressed in interneuron, but also on the tail end of this expression profile, you can also see that there are some expression of cells that are not interneuron. So what are they? They are OLIC1 and GFAP expressing cells. So that means they are oligodendrocytes and astrocytes. So these few cell types are the one that we think for a successful gene therapy, SLC6A1 should be put back there. And then there's another challenge that I would like to highlight is that from Katie Kang's work, is that SLC6A1 is not only mutated, but actually it's stuck in the endoplasmic reticulum. The cell has a intracellular trafficking highway for this protein to be able to express on the cell surface to be functional. So what Katie Kang's lab found is that this SLC6A1, for example, mutation that Maxwell inhibit has this aggregates in the endoplasmic reticulum, not only in astrocytes and also in neurons. But if you look at in the mouse with this mutation, you can see the total amount of protein is actually not different from a normal, from a neurotypical mouse. So that means that these aggregates, it still exist in the cell, in these mutated cells. And when it comes to gene therapy, we are thinking a functional protein that might interact with these aggregates. So will that interference be something that we need to consider? So summarizing this part, we think that there are several things that we need to think about when it comes to gene therapy, a targeted approach. One is that we want to map a therapeutic window. How can we use a systematic way to test which age of this gene therapy would be useful? So we will use a pre-log system collaborating with the Jackson Lab. For brain-wide delivery, we know that there's a widespread expression of SLC6A1. So we might need to brain-penetrating capsid. So that's the vector that can broadly distribute across the brain. Another tricks that we think is that the transgene expression has to be relevant to cell types. So how are we going to do that? We use a native promoter that can drive the endogenous expression pattern of SLC6A1 as a approach to drive cell type-specific restoration. And finally, for checking whether the intracellular trafficking and the interference of this mutated protein might be playing a role in gene therapy, we will also need to test in disease-relevant animal models. So I realize I only have 3 minutes left, so let me just quickly go through what we have done so far in the lab. So to look at systematically looking at the age range of gene therapy, we use this lock-stop approach. Basically, we genetically engineer a stop codon to make the gene not working in mouse. So essentially, it's a knockout situation. But this is a one-way switch that we can use what we call the Cre-Lox recombination to turn on the gene. So in this way, we can systematically test whether introducing a Cre to turn on the gene will restore the SLC6A1 gene expression, but also whether the mouse can be rescued. So this is an ongoing experiment that I believe in Jackson Lab, and Jennifer will talk about her work later in the afternoon. So the other approach that I talked about is that we need a brain-wide delivery. In mouse, we actually have such a tool. The virus that we use, the adeno-associated virus, AAV, that can be injected in the systemic, in the bloodstream of a mouse. And you can see that this virus can actually infect the brain quite efficiently. So the red signal here in the cortex, in the hippocampus and cerebellum, those brain regions that are relevant for SLC6A1, has all good expression of this virus presence. When we co-stain with immunostaining to ask the question, where are those red cells coming from? What are the cell types that express? That we found that's including some of the major inhibitory neuron for albumin interneurons. So that means that this systemic delivery of a brain-wide approach is able to target relevant cells. So what we are using is that we combine with this virus and design a promoter that is restricted, that is endogenous to the SLC6A1 gene. So the idea is that this promoter should drive endogenous pattern of SLC6A1. So that means when the virus goes to a relevant cell, this promoter will be active and driving expression of SLC6A1. When the virus goes to a cell that is not relevant, the promoter will become silent. So does that work in mouse? Yes, the answer seems to be in a handful of animals that we have tried so far. We use a homozygous as benchmark because homozygous is a total knockout of the animal. And this is the EEG spike trains as shown in here. So you can see that the seizure situation in this homozygous mouse. In the total knockout situation, there are a lot of spikes. And we can quantify in here. These are heterozygous that has one copy that's relevant to the patient situations or de novo mutation. And we also see that there are substantially less, but there are conspicuous spikes counts that you can detect. And this is injected with a control virus that does not have the SLC6A1 transgene. Compared to a mouse, also heterozygous, but injected with the SLC6A1 transgene with our construct, we can see that the number of spike trains is significantly reduced, and also the duration, as you can see in this one of the sample trace. So it looks like it is successful in terms of seizure suppression. And we also think about whether there will be some behavior deficits or changes that this might be not safe for this construct. So we typically choose some of the mouse behavior that is relevant for inpatient situation like activity or social behavior or anxiety measures. So we found that basically this SLC6A1 gene therapy in our hand does not show a deficit of behavior. Finally, we're also looking at some of the more basic molecular signaling. So as I mentioned that there are compensatory changes of GABA signaling in the brain in SLC6A1 mutant, which is the case we can actually see that using Western blot, looking at the protein level of this enzyme that makes GABA. So you can actually see that this enzyme seems to be increased. So that fits with the hypothesis that in the SLC6A1 mutant situation, when you have reduced amount of GABA in the presynaptic terminal, the enzyme that produce GABA is compensatorily increased. So the question for us is whether this phenomenon is reversible upon gene therapy. So we now have the tool that we can do that. Before moving away from our model, we also looking at a patient disease, patient mutation related. So this is a collaboration with CADICAN. And instead of using our mouse, we injected CADICAN's person injected SLC6A1 construct into this mouse that has a patient mutation. And we're doing a head-to-head comparison with Dr. Gray's study using a rather early mouse age and for injection. And this is also a seizure count by EEG. As you can see that the seizure without treatment is conspicuously have a lot of spike trains that can be characterized in here. But then after treatment, this seems to be suppressed. So in summary, we think that a novel AAV construct encompassing this native SLC promoter might be able to be effective. And the age that we have testing is actually quite effective even beyond the previously described treatment window. And we are testing now at P55 is about early adulthood of a mouse life. And we're also now working on fine-tuning the SLC6A1 in terms of the dose and the timing. And we are now also with the collaboration testing a disease-relevant disease model and also potentially in human cell models. So I would like to take this opportunity to thank my supervisor, Dr. Alexander Rothenberg, Boston Children's, and a great team of people who is working on this project. And Dr. Gray, and a really great discussion across the years about this gene therapy, and we really learned a lot from him. And also our collaboration with Jackson Lab, with Jennifer and Kelly Kang's work on the disease mouse model. For the clinical translation, because the tool capsid that we use is only usable in mouse, that blood-brain barrier penetrating capsid, we are now collaborating with Dr. Ben Develman at Broad Institute and also his startup company at Futura to bring this therapy to clinic. And of course, I would like to thank Amber for her support and for her inspiration, and also the funding from a few of these entities. Thank you very much for your attention.